Hello again. This is the first in a series of two presentations, A and B, on the introduction to the endocrine system. So this is an integrated approach to human physiology, meaning that now we introduce the endocrine system in this set of two presentations, and then we revisit the endocrine features within the context of organ system physiology and even inter-organ system physiology, for example, cardiovascular and skeletal in exercise, as we proceed through the considerations of the studies in this course. Excuse me. So this chapter then, we consider the function and the purpose of hormones, classification, structure, and synthesis of hormones, pathways of nervous to endocrine regulation, the effects of hormone interactions, and then pathologies of the endocrine system, and just a brief bit about hormone evolution. So we start first with the anatomical summary. Where are hormones produced? So what are the organs or glandular tissues where we find the cells that express the genes for the various hormones. So a range of locations: the pineal gland, the hypothalamus, the posterior pituitary, the anterior pituitary. As we'll see, the posterior pituitary is listed, but it's the site of release, whereas the hypothalamus is the site of the neurons. But from the lecture on the prior chapter, number 6, you understand the type of longer distant neural signaling mechanism, with the cell body and the axon and the site of release. The anterior pituitary gland, the thyroid gland, parathyroid gland, the thymus gland, the heart, and the liver. So this is a primary opportunity to differentiate between an actual gland, where the primary function of the collection of cells in that organ, the anterior pituitary, is a dedication to synthesizing and secreting hormones. Whereas, for example, the heart, obviously it's a circulatory structure. It pumps the blood. That's its primary function. So it is the endocrine cells or an endocrine tissue. It's not an endocrine gland. So if the anterior pituitary produced hormones first and then pumped some blood, and we'd call it a circulatory tissue. I don't know, I'm just joking about that. But it is a highly vascularized organ, so it has the circulatory system highly represented within it, so that it can have the multiple hormones that it produces and releases washed away very quickly to maintain a concentration gradient for the release. So let's consider each of these along the horizontal row for the hormone, the primary targets, and the main effects. So melatonin coming from the pineal gland and the brain and other tissues are the primary targets. It affects circadian rhythms, immune function, and it acts as an antioxidant. The hypothalamus is neural in nature. That's amine in nature. And the trophic hormones are peptides. And the anterior pituitary is the target, and we have release or inhibit of the of pituitary hormones. The posterior pituitary is neural tissue. Oxytocin and vasopressin, or antidiuretic hormone, ADH, both of which are peptides, are the hormones. The target of oxytocin is primarily breast and uterus, and the kidney for the ADH function, but the blood vessels as well for the vasopressin. Obviously, this hormone can increase the vascular tone and contribute to increased blood pressure. But its primary effect, physiological effect, is through the kidney to increase water absorption. The anterior pituitary is glandular tissue, and it is responsible for a set of peptide hormones: prolactin, targeting the breast for milk production; growth hormone, or somatotropin, targeting the liver predominantly for growth factor secretion, or other tissues in general to contribute to growth and metabolism. So we'll talk about this one in more detail as is an example of a more complex endocrine system, neuroendocrine system, because the neural part starts back at the hypothalamus, having these trophic hormones. Corticotropin, or adrenocorticotropin, the ACTH, targeting the adrenal cortex, the outer layers of the adrenal gland, and is responsible for cortisol release. The thyrotropin, or thyroid-stimulating hormone, targets the thyroid gland, of course, and the thyroid hormone synthesis is affected. Now, as we encounter these, or if we encounter individual ones, again separately, we will talk about the specific target cells within the target tissues or target organs, excuse me. So don't think that every cell in the thyroid gland is a target of thyrotropin. Follicle-stimulating hormone and luteinizing hormone target the gonads, both male and female. So quite obviously, there are selected target cells in the gonads. As we know, the gonads have a primary function of gamete production, and to sustain gamete production and reproduction in general, they are endocrine organs and endocrine tissue, and the effect of the follicle-stimulating hormone and luteinizing hormone are on the egg or sperm production, the sex hormone production, and the sex hormone production and the egg or sperm production. So you can see that the follicle-stimulating hormone, like the follicle in the ovary, it primarily is going to affect the gamete production, and that would be accompanied by an effect on sex hormone production. And then it's kind of the reverse for the luteinizing hormone. You don't get a complete development of the gametes without it, but it is predominantly required to get the sex hormone production. So there's subtleties there, and it's obviously a complex system. The thyroid gland, triiodothyronine, I'll push through with that, triiodothyronine, I think I pronounced it a little better there. The thyroxine, this is an amine type of a molecule, and a modified amino acid, excuse me. The A is a modified amino acid. The targets are distributed in many tissues, and there's an impact in general on metabolism, growth, and development. Calcitonin is a peptide, and it is targeting the bone, cells of the bone, responsible for contributing to plasma calcium level maintenance. There's a minimal effect of calcitonin seen in humans. Then parathyroid gland will be responsible for parathyroid hormone, targeting bone and kidney to regulate plasma calcium and phosphate levels predominantly. The thymosin and thymopoietin, both peptide hormones from the thymus gland, will target the lymphocytes and contribute to lymphocyte development. The heart, some of the atrial specialized muscle cells, cardiac muscle cells, also synthesize and secrete a peptide hormone called natriuretic peptide. Well, uresis and natria, natrium is sodium, so we are going to think that you're going to end up with or promote sodium going into the urine. So the target of the kidneys, and indeed increases sodium excretion. So there would be more sodium in the kidneys. Typically, where sodium goes, water goes with it. Angiotensinogen is a peptide hormone from the liver, and then it is acted on by enzymes ultimately to produce angiotensin 1 and then angiotensin 2. That's why it's called angiotensinogen. This is the precursor or prohormone. And its targets are the adrenal cortex and the blood vessels, and will contribute to aldosterone secretion and increases in blood pressure. In other words, the regulation of these physiological functions. Insulin-like growth factors will also be coming from the liver, and they target many tissues. This, of course, is related to the liver targeting many tissues for growth and metabolism. It's through the insulin-like growth factors, and there will be general contribution to growth. Well, we can continue then with these locations and the hormones and the primary targets and the main effects, and we'll just go through each row. The stomach and small intestine, these are individual cells. See, and then the pancreas is referred to as a gland, although it's both exocrine and endocrine gland, so it is an endocrine tissue, not really an endocrine gland, but it's a glandular organ, committed to glandular function. Okay, so the hormones from the stomach and the small intestine, scattered cells, gastrin, cholecystokinin, or CCK, secretin, and other peptide types of hormones. Targets are the GI tract and the pancreas, and they assist in digestion and absorption of nutrients. The pancreas, of course, as we know, is the source of insulin and glucagon, the endocrine pancreas, as well as somatostatin and pancreatic polypeptide. So obviously these are peptide or polypeptide hormones. They target many tissues, and they contribute to the metabolism of glucose, carbohydrates in general, if you want to think of it that way, because all carbohydrates pretty much can go through the glucose point in metabolism, and other nutrients too. The adrenal cortex, again, glandular, aldosterone, cortisol, and androgens. So we talked about the adrenal cortex and the cortisol with ACTH in the prior slide. So the aldosterone is selectively targeting the kidney, while the cortisol and the androgens, the steroid hormones, are targeting many tissues. The aldosterone is sodium and potassium homeostasis, electrolyte balance. There's a stress response for cortisol, and sex drive in females for the androgens from the adrenal cortex. The adrenal medulla, well, we talked about it, epinephrine as well as some norepinephrine, targeting many tissues and contributing to the fight or flight response. The kidney, on a cellular level, different cells in the kidney producing the peptide hormone erythropoietin, 1,25-dihydroxy vitamin D3 or calciferol. I'm sorry, I've forgotten what the S stands for. Oh, down here, steroid. There we go. Look at the legend, right? Targeting the bone marrow and intestine, respectively. The erythropoietin, obviously, red blood cell production, and then calcium homeostasis for the vitamin D. The calciferol, excuse me. And then the skin is where vitamin D3 is finally produced with ultraviolet light. That's an intermediate form of the hormone. It's a precursor to the 1,25-dihydroxy vitamin D3. That's the biologically active form. The testis in the male, they're saying that it is glandular, but typically we think of the male and female gonads as being dedicated to gamete production, and the hormones are a side product required for affecting the organism and contributing to the gamete development. Anyway, in males, the steroid is the general collection of steroids called the androgens, including testosterone, as you know. targeting many tissues, contributing to sustaining sperm production, and contributing to secondary sexual characteristics. Inhibin is a peptide, targets the anterior pituitary to give a negative feedback on FSH secretion. The ovaries in the female, estrogen and progesterone, are the primary steroids categories. Estrogens and progestins and estrogen and progesterone are the major types of steroids within those two categories. So there was the general category of androgens in the male. In the female, there are estrogens, estrogen, which is strictly 17-beta estradiol, is the common estrogen, and progesterone is the common progestin, TIN. So there are general categories that are inclusive of different forms of the steroid molecules. Many tissues are targeted, egg production is sustained, and the secondary sexual characteristics in the female are affected by the estrogens and progestins. The inhibin, again, is a peptide doing the same thing, and negative feedback on the FSH at the level of the anterior pituitary. Relaxin during pregnancy is released. It is a peptide having effects on the uterine muscle to relax it, to contribute to the pregnancy, sustaining the pregnancy. The adipose tissue is a major cellular endocrine tissue, and leptin, adiponectin, and resistin are very potent hormones produced by fat cells, adipocytes, targeting the hypothalamus and other tissues. The hypothalamus specifically to impact food intake and metabolism and reproduction, but as well these other effects are brought about through some of the other tissues that are targeted. But the biggest interest is in the level of the hypothalamus. The placenta, pregnant females only. So again, there are cells that are going to be synthesizing and secreting the female gonadal steroids, estrogens and progesterone, having similar effects, but in this case, sustaining fetal and maternal development. Progesterone promotes gestation. Chorionic somatomammotropin. Let me try that again. Chorionic somatomammotropin. I think I got it. And chorionic gonadotropin. The chorionic gonadotropin targets the corpus luteum to sustain predominantly most importantly, the synthesis and secretion of progesterone to maintain the pregnancy. So the hormone secretion is what's in. And then the chorionic somatomammotropin targeting many tissues to sustain metabolism in the pregnant female. So if you have access to these animations, I encourage you to watch them. I don't want to insert them in these recordings. A recording within a recording is kind of an odd thing, so just stay away from it. So the chemical regulating systems continue in our discussion here. We think of hormones as cell-to-cell communication molecules, as we introduced in the prior set of lectures. They are made in glands or cells, transported to by the blood, so released into the interstitial fluid, diffuses into the blood, and transported and distributed to the distant target tissue receptors. The hormones will activate physiological responses through these receptors. Pheromones are another type of chemical signal, but they are from organism to organism, and strictly speaking, they are organism to organism within the same species, so they are intraspecific chemical messengers. So the butterfly, one species of butterflies, hormones that it secretes is really to have a target effect on another organism of the same species, the same kind of butterfly. Anyway, not such a major concept for understanding human physiology. Although there are some claims and some evidential support, experimental support for human pheromones, we'll just leave it at that. The control of the rates of enzymatic reactions is a typical impact of hormonal functions. The transport of ions or molecules across cell membranes is another example, and then changes in gene expression and protein synthesis, as we had indicated in prior discussions. They exert effects at very low concentrations, so these are potent molecules, so they get diluted in large volumes of blood relative to the concentrations they reach at the site of release with the source cells. Nanogram per milliliter concentrations, as we'll see, are what we find in the plasma, and they can have robust effects on the organism. So they bind to the target cell receptors, so there's a good specificity, and they will have a half-life that indicates the length of its biological activity in general. So we'll talk about half-life at various times throughout the semester. But basically, when a molecule gets into the extracellular fluid, it's not going to stay there forever. Something is going to inactivate it, catabolize it like the liver, and then excrete it like the kidney, and remove it. So it doesn't stay there forever. So there's a way of measuring molecules in the blood, known ones that you add, and you can determine half-lives. And so the half-life values are known for many of the endocrine hormones. The endocrine hormones can be peptide or protein hormones, if we consider their classification, steroids and amines, or modified amino acids, as we had indicated on that first table. So the peptides or proteins can start as a pre-prohormone. Struggled with the angiotensinogen pronunciation initially there to ultimately end up with a description of that. So angiotensinogen is converted by an enzyme to angiotensin 1 is converted by an enzyme to angiotensin 2. So the angiotensinogen is large and it's inactive, and then these enzymes will break it into smaller components, splitting the amino acid bonds, the peptide bonds, and then some of the fragments, typically just one fragment, will become more and more biologically active. Angiotensin 2 is more biologically active than angiotensin 1. Angiotensin 1 has some biological activity, and angiotensinogen, the pre-prohormone, is not active. It's not capable of binding to the receptor. The prohormone is going to be affected by post-translational modifications. This might be phosphorylation more so than what we talked about with the angiotensinogen. The peptide hormone receptor complex would be associated with a signal transduction system. So we're familiar with that from the prior presentation on the chapter 6. Continuing with the classification then, we can make a comparison of peptide, steroid, and amino acid derived hormones. So we see tyrosine derivatives over here on the right, these two columns, catecholamines and thyroid hormones, and then steroid hormones in this middle column, if we consider these two together, and then the peptide hormones over here on the left. So synthesis and storage, peptides made in advance, stored in secretory vesicles. Steroid hormones are lipid soluble, so as soon as they're synthesized, they diffuse away. So the enzymes, the biosynthetic enzyme activity is regulated. So there can be inert precursors that do not diffuse away, readily available for the enzymes to be activated, maybe phosphorylated to turn on, so some signaling mechanism to phosphorylate more of the enzymes, so you convert more of the precursors into the active hormones that diffuse away into the blood and be distributed to target tissues. The catecholamines, this includes epinephrine and norepinephrine that we've talked about a couple times already. They're made in advance, stored in secretory vesicles, and then with the appropriate stimulus, they're released. The thyroid hormones are made in advance, the precursors stored in secretory vesicles. The release from the parent cell, the source cell, it's through exocytosis from the peptide hormones. I said simple diffusion. The site of production is highest concentration, and then the most remote spot from that is lowest concentration. Certainly blood washing through the local vasculature has a lower concentration, so you simply diffuse right into it. Exocytosis for the vesicular release. Simple diffusion, even though there's a storage in secretory vesicles, ultimately there's a cleavage of the lipid-soluble, biologically active thyroid hormones, and then they can simply diffuse into the local blood and be washed away from there. There's a mechanism for these lipid-soluble molecules that can diffuse anywhere to be concentrated in certain areas, either by carrier proteins, as in the plasma, or where you have receptors, such as in populations of cells that express high levels of the receptors. So, for instance, some of the steroid hormones target the nervous system to affect behavior. Think of steroid rage, right? Anabolic steroid abuse leads to changes in the nervous system. Well, it places in the nervous system rage centers, like the amygdala. You have a high concentration or a collection of neurons that express the receptor gene for the steroids at high levels, and so those molecules will be binding and unbinding the steroid hormone in that local area, and will just kind of cause a local reserve, kind of like a sponge would hold water in more location out in your yard than the water just being in a mud puddle. Kind of a thing like that. So anyway, there are carrier molecules, transport proteins in the plasma that help to get a concentration into the plasma to be delivered to target sites, and then the cells that need a lot of it nearby will help sustain a local concentration by expressing the receptor gene. In addition to, making themselves responsive to the hormones, being target cells. Transport in the blood, it's dissolved in the plasma. Steroid hormones bound to proteins, and then dissolved in the plasma. The half-life is short for peptide hormones. They get gobbled up by enzymes that degrade proteins pretty fast. The steroid hormones, of course, because they're bound to carrier proteins or held close to the cells that need them to be responsive, so they'll last a little bit longer. Same short for catecholamines. You don't want epinephrine to stay in your blood for a long period of time, and make your heart just burst in your chest. You want to have a short-term response and then have it terminated. And then you have thyroid hormone that hangs around for quite a long time, so long half-life. The location of the receptor for the water-soluble hormones, it's always going to be on the cell membrane. For the lipid-soluble hormones, the cytoplasm or the nucleus, but sometimes in the membrane too. So we saw that in the Chapter 6 presentation. Thyroid hormones, specifically, the receptors will be located right in the nucleus, bind the thyroid hormones, and then bounce around and end up sticking to particular DNA nucleic acid sequences on selective genes, target genes, and have effects on gene expression. Whereas the cytoplasmic ones for the steroid hormones has to be ligated, then it can get into the nucleus and do that. The response to the receptor ligand binding, you'll have activation of second messenger systems that may activate genes to peptide hormones. Still activation of genes, as I said, changing transcription rates, and then thereby having impact on translation, the production of protein. And it may have non-genomic effects as well. Some are well established, don't understand exactly how they are produced, but they're reproducible. So we know that there are some non-genomic effects on proteins within the membranes, giving independent responses independent of changes in gene transcription and translation. So the water soluble is the activation of second messenger systems, and then of course the binding to the receptor in the nucleus is going to have an immediate direct effect on gene expression, so the transcription rate, and thereby impact on translation. So general target response for the peptide hormones: modification of existing proteins and induction of new protein synthesis. And the effects on transcription, obviously new protein synthesis. The catecholamines on the cell membrane receptor activating second messenger is going to modify existing proteins. And then obviously the thyroid hormones right at the nucleus going to affect new proteins. Examples of peptides: insulin, parathyroid hormone; estrogen, androgen, cortisol for the steroids; epinephrine and norepinephrine, as I mentioned, for the catecholamines; and then thyroxine, the tetraiodothyronine, or T4, is a prime example. Well, now we'll consider the peptide hormone synthesis, packaging, and release, and we'll go through a series of slides that will present each step in an additive form. So we'll go to this slide. First, the messenger RNA on the ribosomes binds amino acids into a peptide chain called a pre-pro hormone. The chain is directed into the ER lumen by a signal sequence of amino acids. Now, the angiotensinogen is an exception to what we're going to consider here. The cells secrete the angiotensinogen right into the extracellular fluid. It gets into the blood, and then endothelial cells or cells in the kidney will produce the enzymes that modify the pro hormone. Anyway, so that's a good example sometimes, and then not such a good example when you're thinking of it in this way. So let's get back here. We've got messenger RNA binding to the ribosomes, and then you get translation of a full length representing the the mRNA sequence. Then there's going to be enzymes processing mechanisms in the endoplasmic reticulum to enzymatically cleave portions of this molecule. The pre-pro hormone specifically has what's called the signal sequence. This signal sequence is on all proteins that are translated at the endoplasmic reticulum, and it would have been presented in the see, for this much protein being in here, this messenger RNA. Anyway, I guess this is reading it from this end to that end, but usually it's anyway. Let's not get caught into the specifics here. But anyway, this is represented somewhere in the messenger RNA in the first part that is translated. The first part of the protein that comes off is the signal sequence for all proteins that are going to end up being processed by the endoplasmic reticulum. So after the cleavage of the signal sequence, then you have the pro hormone, which would actually be the angiotensinogen if I was to be strictly specific about it. Then there is the prohormone getting passed from the ER to the Golgi complex. So this would be very familiar to you, all the way through the Golgi without any additional enzymatic cleavage, and then finally being extruded on the other end of the Golgi apparatus into a secretory vesicle that also will have packaged into it certain enzymes. And the prohormone is also in there. They bud off the Golgi. The enzymes are going to be the ones that will further process or cleave or chop the prohormone into one or more active peptides plus the additional peptide fragments. So that if you can harvest these vesicles, these later, more mature secretory vesicles, you would find the peptide fragments in there as well. Now, in angiotensinogen, this doesn't happen. This form, the prohormone, gets released, secreted, and then it gets into the blood, and then that's where these other enzymes are that will activate it. So it's a very unique and interesting endocrine system. And then with the appropriate signal, of course, you get the release. Not only of the active hormone, but of the peptide fragments as well. And in some cases, the peptide fragments have effects too. And of course, I like this because we see the release into the interstitial fluid compartment first, and then diffusion down the concentration gradient into the plasma. So you have to have the blood flow to keep washing this away, so you keep a lower concentration here at number 6 than you have here at number 5. So the peptide hormone synthesis, packaging, and release continues when we look at this type of linear representation or illustration of a pre-pro hormone. In this case, for thyrotropin releasing hormone, we can see it's a tripeptide. So it's actually repeated in the case of humans 6 times throughout the length of the pre-pro hormone. So the pre-pro TRH, thyroid releasing hormone, is 242 amino acids, and then it gets enzymatically cleaved to the hormone and other peptides, and there's 6 TRH tripeptides, 3 amino acids each, within each pre-pro hormone. So you get a lot of bang for your dollar, right? And then, of course, don't forget about the signal sequence, since this is the pre-pro hormone shown here in that pinkish-orange color. The pro-opiomelanocortin system is a bit different. We're not seeing the pre-pro hormone. We're starting with the pro hormone, so the signal sequence has already been cleaved in this case. There's additional processing into 1, 2, 3, 4 fragments: ACTH, lipotropin, endorphin, plus another fragment. And then there's other cases, such as the proinsulin being processed. First, there are at least these 4 cysteine residues. Cysteine amino acid has the sulfides and is involved in disulfide bonds. These disulfide linkages contribute to the tertiary structure of this protein, and then that is the proinsulin. Then there are enzymatic cleavage sites at these 2 locations that release this pinkish color from the proinsulin, called the C-peptide, and then the active insulin molecule. So this is kind of an interesting final product of a linear polypeptide prohormone. So, we have a great need with all the diabetes in the world for insulin. So when they first started putting the insulin gene into bacteria, there was a lot of doubt that they would be able to generate this type of insulin. But they ultimately got it worked out. And biotechnology is the primary source of insulin now for treating human diabetes, and animals too, diabetic animals. If you have a pet, you can get the insulin for them too. Whereas they used to have to just get it from slaughtered pigs and things like that to isolate it from their plasma. So the peptide hormone receptor complex, then, it's a surface receptor. The hormone binds. There's enzyme activation. We'll get opening of channels, second messenger systems, and cellular responses. So let's take a look at this again. And yes, we did just see it in Chapter 6. So we're just going to go through. So the membrane receptors and signal transduction for peptide hormones. So we're picturing these as peptide hormones. We recognize the tyrosine kinase, the adenylate cyclase, the G coupling, and then an ion channel, second messenger to phosphorylate proteins and get the response. The steroid hormones will be a bit different, and we'll take a little bit closer look at them in this presentation. So they are cholesterol derived, they are lipophilic and can enter the target cell, freely diffuse through the lipid membranes. The cytoplasmic or nuclear receptors will be present. Mostly we find nuclear receptors, but there are some examples of cytoplasmic. The reason they're more likely to get right to the nucleus first is that they're targeting DNA once they're ligated, so it's obvious that such a system would have evolved. So they activate DNA for protein synthesis. See, this isn't necessarily true. If you think of negative feedback at the anterior pituitary, the gonadal steroid hormones are going to inactivate DNA so that you get less secretion. That's what negative feedback should do, right? But anyway, they alter transcription rates appropriately and have impacts on protein synthesis. In the case of the anabolic steroids, where you're using a synthetic exogenous testosterone-like or androgen-like molecule, it increases the DNA activity for the skeletal muscle proteins, so you get bigger, bulkier muscles. So that's an exaggerated example of it, right? Anyway, the slower acting, longer half-life of these steroid hormones was mentioned on a prior table, and the examples as well, cortisol, estrogen, and testosterone. Well, the steroid hormone structures are considered in this slide. So here we see the cholesterol. It's the parent compound. So enzymes will modify it to produce in the adrenal cortex aldosterone and cortisol, and in the ovary estradiol, which is an estrogen. So I want to draw your attention to these molecules. You should be able to recognize and identify each of them. So estradiol, a diol, it's an alcohol, meaning that is an OH group. So diol, meaning there's two of them. So there's two OHs. But let's go back to the cholesterol molecule and just recognize the basic ring pattern. There's an A, B, C, D ring pattern, 3 hexagons, right, and 1 pentagon. So that's true in the case of all of these. So as you're scanning, if you see any of these images, that's the thing to do, is go A, B, C, D. It is a sterol. And if it doesn't have a lot of carbons coming off the top of the little house here, the pentagon, then it's going to be one of these hormones. So you can see there's that long carbon chain out there with these 2 methyl groups hanging off the final carbon. So that's the cholesterol. Cholesterol, OL, so there's 1 OH group. And then kind of look for double bonds as you're going through the ring structures. So ring A, B, C, D. So all the hormones are going to have the double bond in the A ring, and estradiol is going to have not only 2 OHs, but 3 double bonds in the A ring. Cortisol just has 1, and a double bond O on that A ring, one of the carbons there, and I'm not going to take you through the numbering system or stuff like that. So what's the difference between aldosterone and cortisol? And then ultimately you'll see testosterone and progesterone, and I'll want you to try to recognize all of these. So what is the difference between aldosterone and cortisol? So we have a methyl there, hydroxyl group there, and we've got a CH and a double bond O for the aldosterone, aldo, so we got an extra O up there. Otherwise, we're almost the same, except that cortisol, OL, there's an OH group on the top of the little house. Right? So make those connections so that you would be able to identify all of these, or any of them, any one of them, in a testing situation. So the steroid hormone actions we're going to consider in the next series of slides in 5 steps. So there's the protein carrier. Albumin is a common protein carrier found in the plasma. And there's some specific hormone carrier proteins as well. But for now, we'll just think of a protein carrier with the the steroid hormones spending more time stuck to it than free in the blood. So you might be aware, like with thyroid hormone, you can do a measurement of free thyroid versus bound. So they should have one little yellow ball here that would be free and then bound. And there's a ratio of free to bound that depends upon how much hormone there is versus protein carrier that's available. You know, I don't like to leave you with these simple things when you need a better understanding of it. Either way, the free becomes available to diffuse away, right? So the most hydrophobic steroids are bound to plasma protein carriers. Only unbound hormones can diffuse into the target cell. Well, they should have shown the free form in the plasma as well, but anyway, the free would be here, and it is the one that is free, available to diffuse down its concentration gradient. It's a higher concentration gradient in the plasma. Why? Because you have the carrier proteins that bind it, keep it held close more frequently. When there isn't a binding site, it just diffuses away, simple diffusion in all directions. As it's simply diffusing, it looks like it here that it goes, oh, I know where to go, right there. Well, it doesn't. It's diffused. The free is diffusing everywhere, and that which encounters, it binds to and recepts the binds to and stays on the cytoplasmic receptor would be this one. So if a molecule missed that and diffused here through the nucleus, it might encounter the nuclear receptor. So they always make it look like these molecules know where they need to go, and they just, you know, kind of walk their way along or fly there. I don't know what it's supposed to be, but they diffuse in every which direction, and there's a bunch of yellow circles, and they go in every which direction, and those that get here bind to that or have an opportunity to bind to it, because they're all randomly bouncing around, right? And then the nuclear receptor here. So they bind to the receptors, either cytoplasm or nucleus, or they can bind to the cell surface receptor and have a rapid response. The cytoplasmic receptors, once they're ligated, change their conformation, and then the guard proteins at the nuclear pores say, okay, you have permission now to enter the nucleus. Either way, once they're in the ligated form, the steroid hormone receptor complex can bind to DNA and affect the gene transcription. If it's going to activate genes, remember, it can activate or inactivate. Doesn't have to be just one or the other. It can be a little bit of both. But typically with one gene, a particular steroid like estrogen bound to its receptor, the estrogen receptor, will have a particular effect. But remember, these are all over. It's not just one of them. You know, they're showing two here and then one there, so they're simplifying it too much. It's all concentration dependent. Then the transcription would produce the mRNA, so the levels of mRNA would be affected up or down. And then as well, translation would be affected up or down. Then the new protein levels would be up or down correspondingly as well. So if this was the anterior pituitary and we're talking about negative feedback on an anterior pituitary hormone, then you'd want to inhibit the expression of the gene there because testosterone levels are too high, right? So if you go back to the example of a person taking an anabolic steroid that looks like testosterone, well, then it gets the anterior pituitary and shuts down the FSH and the LH genes, so you no longer produce them. And what happens to their gonads? Yeah, they shrivel up and stop producing anything, right? Because the exogenous hormone is producing an inhibition, a negative feedback on the transcription of the pituitary hormones, the reproductive pituitary hormones, the LH and FSH that we saw in the previous table. So if they're careful about how they do it and they stop at an appropriate time, then the system can recover. Some individuals take so much and for such a long period of time, these are permanently shut off or at such a low level that they then have to be supplemented the rest of their life with exogenous testosterone. The amine hormones then, we look at the features of those. So they're derived from one of two amino acids, tryptophan or tyrosine. So these have a ring structure, producing thyroid hormones or the catecholamines, epinephrine, norepinephrine. Dopamine is included in there. Actually, dopamine can be converted to norepinephrine, and then norepinephrine enzymatically can be converted to epinephrine. So the derivatives of tyrosine, here we see the typical amino acid, the COOH, and the amine group. So this is the amino and carboxy groups that make it an amino acid. Then this is the R group. And the tyrosine has that single ring, a phenolic ring with the 3 double bonds in it. Phenol, OL, OH on it, right? If you see the iodines on there, so obviously 2 tyrosines get stuck together at the O here, and then iodine is added, and then there's cleavage of, well, they're not 2 of them at this end, but one with the OH and then the other where all the carbons are taken off gets stuck to that oxygen. Anyway, count the I's. 1, 2, 3, 4. You'd be surprised how many people have on exams they'll pick triiodothyronine for this, and it's like, but there's 4 I's there. Anyway, tetra means 4, and tri means 3. So count the I's, right? Obviously, there's 2 phenol rings, so it's not tyrosine. It's not the ABCD rings of the sterols. And then here you see the catecholamines. So you don't have the 2 phenol rings stuck together, so there's just 1. So you might confuse them with the tyrosine, but look, there's 2 hydroxy groups on them. Plus, they're decarboxylated. The carboxyl groups are gone. See, so the carboxylic acid, making this an amino acid, the carboxylic acid has been removed. So there's a dopa decarboxylase enzyme that does that. It produces the dopamine. So that's dopamine. Then norepinephrine, look, it's just an OH group rather than a hydrogen on this carbon closest to the ring. And then epinephrine has a methyl group on there. The enzyme that converts this to this is called phenyl, so the phenol group, phenylethanol, ethanol, amine, N-methyltransferase. So phenylethanol-amine N-methyltransferase transfers a methyl group there and converts norepinephrine to epinephrine. So I know that's a lot of details, but I'd like to give you a bit of insight, helps you understand naming nomenclature of enzymes and things like that. But most importantly, slight differences in the molecules. It's just an OH here rather than an H. And then add a methyl group and look at the differences you can get between dopamine, norepinephrine, and epinephrine biologically. Just phenomenal. And subtle differences between testosterone, estrogen, and progesterone, and you get dramatically different biological effects. So that's the point to be considered and understood, locked into your brain from that discussion, other than saying I need to come up with a way to recognize them if I see them on a performance assessment exam. So let's think about the endocrine reflex pathways then. We consider these in the chapter 6, stimulus, afferent signal, integration, efferent signal, the hormone, physiological action, and then some way of terminating or regulating the system altogether. So here we see it. Oops. Sorry about that. This is obviously our last slide. The endocrine reflex pathways for insulin release are shown on this slide. So insulin is related to plasma glucose levels. So we are looking at plasma glucose levels being high or increasing up here and decreasing or being low over here. Okay, so we eat a meal, and there will be stretch receptors in the digestive tract that have an afferent or sensory neuron that will signal through a neurotransmitter to an efferent neuron in the CNS that will innervate the pancreas and trigger the release of insulin. But there's also a direct effect if you eat a meal, the digestion, or if it's just raw sugar in there or glucose, then you get an increase in blood glucose levels. And these have the direct effect on the pancreas for the endocrine reflex. Then the insulin synthesis and secretion is controlled. If you have high blood glucose, then the pancreatic cells are going to release insulin, increasing insulin concentration locally, and then it's washed away and increasing it in the blood and at the target tissue sites, especially and importantly the skeletal muscle and the liver, where that will act through a receptor to increase glucose uptake and utilization. Should be to store as glycogen first, and then as lipids, or to be converted to amino acids if needed. And then this will be like a sponge, the skeletal muscle and the liver, to suck the glucose out of the blood, right, and bring the blood glucose down. And then that will be a negative feedback effect, actually coming in here more so to just alleviate the drive at the pancreas. I gotta stop recording.